MCQs on microscopy. Which microscopy technique uses the principle of constructive and destructive interference of light waves to enhance contrast in unstained specimens? A. Bright field microscopy. B. Phase contrast microscopy. C. Fluorescence microscopy. D. Polarized light microscopy. Explanation: phase contrast converts phase shifts of light passing through transparent specimens into intensity differences, making unstained samples visible. Answer: B. Phase contrast microscopy. Which of the following is the main principle of confocal laser scanning microscopy, CLSM? A. Use of polarized light. B. Use of a pinhole aperture to eliminate out-of-focus light. C. Differential staining of structures. D. Electron beam interaction with specimen. Explanation: CLSM uses a focused laser and pinhole to capture only in-focus light, enabling 3D reconstruction of thick specimens. Answer: B. Use of a pinhole aperture to eliminate out-of-focus light. Which microscopy technique is most suitable for visualizing surface topography at atomic resolution? A. Transmission electron microscopy, TEM. B. Atomic force microscopy, AFM. C. Scanning electron microscopy, SEM. D. Phase contrast microscopy. Explanation: AFM uses a sharp tip that scans surfaces and measures forces, providing atomic level 3D surface imaging. Answer: B. Atomic force microscopy, AFM. In fluorescence microscopy, which phenomenon allows specific molecules to emit light of longer wavelength after excitation? A. Reflection. B. Refraction. C. Fluorescence emission. D. Polarization. Explanation: fluorophores absorb light at a shorter wavelength, excitation, and emit at a longer wavelength, emission, enabling specific labeling. Answer: C. Fluorescence emission. Which microscopy type exploits differences in refractive index and birefringence of biological samples? A. Polarized light microscopy. B. Dark field microscopy. C. Confocal microscopy. D. Fluorescence microscopy. Explanation: polarized microscopy enhances contrast in anisotropic specimens, example crystalline structures and muscle fibers. Answer: A. Polarized light microscopy. What is the resolution limit of a typical transmission electron microscope, TEM? A. 200 nm. B. 20 nm. C. 0.2 nm. D. 2 μm. Explanation: TEM provides atomic scale resolution, 0.2 nm, because electrons have very short wavelengths compared to visible light. Answer: C. 0.2 nm. Which microscopy technique enhances edges and fine details by using oblique light illumination? A. Differential interference contrast, DIC, microscopy. B. Dark field microscopy. C. Phase contrast microscopy. D. Fluorescence microscopy. Explanation: dark field blocks direct light, allowing only scattered light to form the image, making edges and small structures visible. Answer: B. Dark field microscopy. Which principle allows scanning electron microscopy, SEM, to generate detailed 3D-like surface images? A. Detection of transmitted electrons. B. Detection of secondary electrons emitted from surface atoms. C. Measurement of light interference patterns. D. Detection of X-rays from atoms. Explanation: SEM scans with an electron beam and detects emitted secondary electrons, producing high-resolution 3D surface images. Answer: B. Detection of secondary electrons emitted from surface atoms. Which microscopy technique is based on tunneling current between a sharp tip and the sample surface? A. AFM. B. STM. C. SEM. D. TEM. Explanation: Scanning tunneling microscopy, STM, measures tunneling current between tip and conductive surface, allowing atomic resolution imaging. Answer: B. STM. Cryo-electron microscopy, Cryo-EM, is advantageous over conventional TEM because A. It uses X-rays instead of electrons. B. It preserves samples in a near-native hydrated state. C. It requires chemical staining of samples. D. It only works for metallic specimens. Explanation: Cryo-EM rapidly freezes specimens, avoiding artifacts from dehydration or staining, ideal for proteins and viruses. Answer: B. It preserves samples in a near-native hydrated state. Which microscopy technique is most suitable for real-time visualization of live cells with minimal photo damage? A. Light sheet fluorescence microscopy. B. Bright field microscopy. C. Scanning electron microscopy. D. Polarized microscopy. Explanation: Light sheet illuminates only a thin optical section, reducing phototoxicity and allowing long-term live cell imaging. Answer: A. Light sheet fluorescence microscopy. The resolving power of a microscope is primarily dependent on A. Magnification of objective lens. B. Wavelength of illumination source and numerical aperture. C. Size of the specimen. D. Thickness of the cover slip. Explanation: Resolution equals lambda/2 Na. Shorter wavelength and higher Na improve resolution. Answer: B. Wavelength of illumination source and numerical aperture. Which technique is used for mapping chemical composition on surfaces using electron microscopy? A. Bright field microscopy. B. EDS coupled with SEM. C. Polarized microscopy. D. Phase contrast microscopy. Explanation: Energy dispersive X-ray spectroscopy, EDS, detects characteristic X-rays emitted from atoms excited by electron beams. Answer: B. EDS coupled with SEM. Which microscopy type is based on interference of two beams of polarized light to produce pseudo-3D images? A. Light microscopy. B. SEM. C. TEM. D. Dark field microscopy. Explanation: Differential interference contrast uses polarized beams to enhance contrast, giving a 3D-like appearance. Answer: A. Light microscopy. Which of the following is a limitation of fluorescence microscopy? A. High phototoxicity and bleaching of fluorophores. B. Low specificity of labeling. C. Poor surface imaging. D. Inability to resolve structures smaller than 0.1 nanometers. Explanation: Fluorescent dyes can degrade under prolonged excitation, causing reduced signal and cell damage. Answer: A. High phototoxicity and bleaching of fluorophores. Which microscopy method is best suited for observing nanostructures below the diffraction limit of light, 200 nm? A. Bright field microscopy. B. Super resolution microscopy, STORM, POM, SIM. C. Phase contrast microscopy. D. Polarized microscopy. Explanation: Super resolution bypasses diffraction limit, achieving resolution up to 20 to 50 nanometers. Answer: B. Super resolution microscopy, STORM, POM, SIM. What is the principle of scanning probe microscopy, SPM? A. Transmission of electrons through thin samples. B. Light scattering and diffraction. C. Interaction of a sharp probe with sample surface forces. D. Fluorescent emission from labeled molecules. Explanation: SPM includes AFM and STM, which rely on probe-sample interactions for nanoscale imaging. Answer: C. Interaction of a sharp probe with sample surface forces. Which microscopy is most useful for visualizing microtubules labeled with GFP in live cells? A. SEM. B. Fluorescence microscopy. C. Polarized light microscopy. D. TEM. Explanation: fluorescent proteins like GFP allow live cell visualization of cytoskeletal structures using fluorescence microscopes. Answer: B. Fluorescence microscopy. Which type of microscopy allows measurement of molecular interactions using Forster resonance energy transfer, FRET? A. SEM. B. Fluorescence microscopy. C. TEM. D. Dark field microscopy. Explanation: FRET detects energy transfer between two nearby fluorophores, revealing molecular interactions in real time. Answer: B. Fluorescence microscopy. What is a key limitation of SEM compared to TEM? A. Lower magnification range. B. Inability to image surface structures. C. Poor vacuum requirement. D. Inability to visualize internal ultrastructure. Explanation: SEM only scans surfaces, whereas TEM penetrates thin sections to reveal internal ultrastructural details. Answer: D. Inability to visualize internal ultrastructure. In transmission electron microscopy, TEM, the function of the electromagnetic condenser lens is to: A. Form the final image. B. Focus the electron beam onto the specimen. C. Increase magnification of the objective lens. D. Detect secondary electrons. Explanation: the condenser lens system in TEM focuses the electron beam into a fine probe on the specimen for uniform illumination. Answer: B. Focus the electron beam onto the specimen. In cryo-electron tomography, Cryo-ET, 3D reconstruction is achieved by: A. Sectioning the specimen physically. B. Rotating the specimen and capturing multiple 2D projections. C. Using multiple detectors. D. Scanning in XY plane with atomic force. Explanation: Cryo-ET collects 2D images at different tilt angles to computationally reconstruct 3D ultrastructure. Answer: B. Rotating the specimen and capturing multiple 2D projections. Which microscopy technique achieves atomic resolution through measurement of quantum tunneling currents? A. AFM. B. STM. C. SEM. D. TEM. Explanation: scanning tunneling microscopy measures tunneling current between a conductive tip and the sample surface, giving atomic scale resolution. Answer: B. STM. In atomic force microscopy, AFM, which mode detects van der Waals forces without contact? A. Contact mode. B. Tapping mode. C. Non-contact mode. D. Force spectroscopy mode. Explanation: non-contact AFM scans above the surface, detecting weak van der Waals forces, ideal for delicate biological samples. Answer: C. Non-contact mode. Which microscopy type breaks the diffraction limit by controlling fluorophore excitation using two synchronized laser beams? A. SIM. B. POM. C. STORM. D. STED. Explanation: stimulated emission depletion, STED, microscopy uses a depletion laser to restrict fluorescence to sub-diffraction volumes, achieving 20 nm resolution. Answer: D. STED. What limits the resolution of light microscopy according to Abbe's equation? A. Lens curvature. B. Numerical aperture and wavelength. C. Depth of focus. D. Light source intensity. Explanation: resolution equals lambda/2 Na. Shorter wavelength and higher Na improve resolving power. Answer: B. Numerical aperture and wavelength. The electron wavelength in a TEM operating at 200 kV is approximately A. 2.5 nanometers. B. 0.025 nanometers. C. 0.0025 nanometers. D. 0.25 nanometers. Explanation: accelerated electrons at 200 kV have de Broglie wavelength around 0.0025 nanometers, much shorter than visible light. Answer: C. 0.0025 nanometers. In differential interference contrast, DIC, microscopy, which optical component splits polarized light into two beams? A. Phase plate. B. Wollaston or Namarsky prism. C. Pinhole aperture. D. Dichroic mirror. Explanation: these prisms separate polarized light into two beams that recombine to create interference and pseudo-3D contrast. Answer: B. Wollaston or Namarsky prism. Which microscopy is most useful for mapping atomic composition with sub-nanometer spatial resolution? A. SEM. B. STEM-EDS. C. AFM. D. DIC. Explanation: scanning transmission electron microscopy combined with energy dispersive X-ray spectroscopy gives elemental mapping at atomic resolution. Answer: B. STEM-EDS. In scanning electron microscopy, backscattered electrons primarily provide information about A. Surface roughness. B. Atomic number contrast. C. Specimen thickness. D. Fluorescence intensity. Explanation: backscattered electron yield increases with atomic number, giving compositional contrast. Answer: B. Atomic number contrast. Which detector in SEM collects secondary electrons emitted from the sample? A. Everhart-Thornley detector. B. Scintillation detector. C. CCD camera. D. Faraday cup. Explanation: this detector captures low-energy secondary electrons to produce topographic images. Answer: A. Everhart-Thornley detector. Which microscopy technique combines optical and AFM systems to obtain both topographic and fluorescence data? A. Correlative light electron microscopy, CLEM. B. Near-field scanning optical microscopy, NSOM. C. STORM. D. Superconducting probe microscopy. Explanation: NSOM uses a sharp probe to collect optical signals in the near field, exceeding the diffraction limit and combining topographic and optical data. Answer: B. Near-field scanning optical microscopy, NSOM. In transmission electron microscopy, sample thickness must be A. 50 m. B. 50 to 100 nm. C. 1 m. D. 1 mm. Explanation: TEM requires ultra-thin sections to allow electron transmission without significant scattering. Answer: B. 50 to 100 nm. The image contrast in phase contrast microscopy arises due to A. Absorption of light. B. Phase difference converted to intensity differences. C. Diffraction of light. D. Polarization of light. Explanation: phase shifts of light waves passing through transparent specimens are converted into visible brightness variations. Answer: B. Phase difference converted to intensity differences. Which super-resolution method uses sequential activation and localization of individual fluorophores? A. SIM. B. PALM/STORM. C. STED. D. CLSM. Explanation: PALM, photo-activated localization microscopy, and STORM, stochastic optical reconstruction microscopy, build super-resolved images by localizing single molecules over time. Answer: B. PALM/STORM. Electron energy loss spectroscopy, EELS, in TEM provides information on A. Atomic number only. B. Elemental composition and electronic structure. C. Surface roughness. D. Phase contrast. Explanation: EELS measures the energy lost by electrons passing through a sample, revealing atomic bonding and oxidation states. Answer: B. Elemental composition and electronic structure. Structure. What is the typical accelerating voltage range for SEM imaging? A. 1 to 5 V. B. 1 to 30 kV. C. 100 to 500 kV. D. 0.1 to 1 MV. Explanation: SEM commonly operates between 1 to 30 kV, lower voltages are used for surface-sensitive imaging. Answer: B. 1 to 30 kV. In fluorescence microscopy, the dichroic mirror serves to A. Focus excitation light. B. Separate excitation and emission wavelengths. C. Reduce background noise. D. Increase numerical aperture. Explanation: the dichroic mirror reflects excitation light toward the specimen while transmitting emitted fluorescence to the detector. Answer: B. Separate excitation and emission wavelengths. The resolving power of an electron microscope is enhanced mainly due to A. Shorter wavelength of electrons. B. Higher numerical aperture lenses. C. Stronger illumination intensity. D. Vacuum environment. Explanation: electrons have much shorter wavelengths, 0.002 nm, giving extremely high resolving power compared to light microscopy. Answer: A. Shorter wavelength of electrons. Which microscopy technique allows dynamic imaging of live cell membranes at nanometer resolution using a probe? A. TEM. B. AFM in liquid mode. C. SEM. D. Light microscopy. Explanation: AFM can operate in physiological conditions, liquid mode, to study live cell membranes and biomolecular interactions. Answer: B. AFM in liquid mode. In bright field microscopy, the condenser's primary function is to A. Increase resolution. B. Focus light onto the specimen. C. Increase magnification. D. Reduce spherical aberration. Explanation: the condenser directs light precisely on the specimen to optimize illumination and contrast. Answer: B. Focus light onto the specimen. Which is not a correct pairing of microscopy and detector type? A. TEM, fluorescent detector. B. SEM, secondary electron detector. C. Fluorescence microscopy, photomultiplier tube. D. AFM, laser deflection sensor. Explanation: TEM uses scintillator and CCD cameras, not fluorescent detectors. Answer: A. TEM, fluorescent detector. Which microscopy technique visualizes mechanical properties, like stiffness, of biological samples? A. AFM. B. SEM. C. TEM. D. Phase contrast microscopy. Explanation: AFM can perform force spectroscopy to measure elasticity, adhesion, and stiffness at nanometer resolution. Answer: A. AFM. In electron microscopy, the use of heavy metal stains, example osmium tetroxide, is primarily to A. Reduce sample charging. B. Increase electron scattering contrast. C. Enhance fluorescence. D. Neutralize sample charge. Explanation: heavy metals scatter electrons strongly, improving image contrast of cellular structures. Answer: B. Increase electron scattering contrast. Which microscopy method combines fluorescence with electron microscopy for precise correlation of structure and function? A. CLEM. B. CLSM. C. AFM. D. DIC. Explanation: correlative light and electron microscopy, CLEM, integrate fluorescent labeling with ultrastructural imaging. Answer: A. CLEM. In TEM, spherical aberration is corrected by A. Using magnetic prisms. B. Using CS corrector lenses. C. Reducing accelerating voltage. D. Using immersion oil. Explanation: spherical aberration correctors improve image sharpness and resolution by compensating for lens imperfections. Answer: B. Using CS corrector lenses. Which optical phenomenon allows total internal reflection fluorescence, TIRF, microscopy to selectively excite fluorophores near the cover slip? A. Interference. B. Evanescent wave excitation. C. Reflection. D. Refraction. Explanation: TIRF generates an evanescent field that excites fluorophores only within 100 nm of the glass-water interface. Answer: B. Evanescent wave excitation. In TEM imaging, the diffraction contrast mainly arises from A. Incoherent scattering. B. Elastic scattering from crystal planes. C. Secondary emission. D. Phase interference of light. Explanation: diffraction contrast reveals crystallographic details based on electron diffraction patterns from lattice planes. Answer: B. Elastic scattering from crystal planes. In super-resolution microscopy, what is the main principle behind SIM, structured illumination microscopy? A. Random activation of fluorophores. B. Interference of patterned light with sample structure. C. Depletion of excited fluorophores. D. Tunneling current detection. Explanation: SIM uses patterned illumination and computational reconstruction to double the resolution of conventional fluorescence microscopy. Answer: B. Interference of patterned light with sample structure.